Welcome back, everyone. Thanks for taking a little break with us. Satish is going to continue on with how to use Snakemake, so I'm going to just turn it right over to Satish.
Okay, thank you, Tina. Welcome back, everyone. So in the part one, we saw just the basics of Snakemake, and we saw some of the features. Well, now let's try to run an end-to-end RNA sequencing analysis workflow, Snakemake workflow within this environment. So here you can see some introduction about the analysis. This RNA sequencing analysis is of 3 strains, 1 test sample and 2 controls. And this is not a typical workflow that you might be working on, because it's completely self-contained workflow where the data is generated from the NCBI SRA database and it generates PDF files. But it's a good place to start learning, building up the workflows. So this is the rule graph for the Snakemake workflow for RNA sequencing analysis. And just by the rule names, maybe you can make out the different steps that are involved in the RNA sequencing analysis, where you would have to get the reference genome FASTA file, index the reference genome, align to the genome the sequence reads that you download from the NCBI SRA database by accession. Here is a separate rule for performing quality control on these samples by using FastQC and MultiQC. You align the reads to the genome, sort the BAM files, get the gene annotation genome file GFF, and using the annotation file and the BAM file, you generate the count table, which will then be used for further downstream analysis. And as you can see, this Snakemake workflow has this particular workflow. What we will do in the rest, I think like now the rest of the webinar would be to run the workflow, take time to look at the Snakemake snake file that they have in used in generating this workflow. And towards the end, I also want to mention, take time to discuss about how you can generate your own Jupyter Lab notebooks like these within the CyVerse discovery environment. Okay, so to run the RNA sequencing analysis workflow, I would request you to go back to the Read the Docs webpage. And first, we are going to git clone the workshop repository and change directories into a particular folder called the Docker. And then we are going to move to the development branch of this particular project, because they're still updating the contents of the workshop. So we will move to the developing branch just to be on with the latest updates. So I would request you to go ahead and copy these and paste them into your terminal within the Jupyter notebook. So the first command will git clone the workshop reproducible research folder. And then we will change directories within the workshop reproducible research into the Docker folder. And here within the Docker folder, you can see there are a bunch of files, but there's also a snake file, which is the snake file for RNA sequencing. You can open that and you can see all, this is a very well-written Snakemake workflow, I have to say. We will go over this once we set up the workflow for execution. Okay, but before first we set up the workflow for execution, let us generate the rule graph within for this workflow. So to generate the rule graph, I'll be using the -- rule graph option. And I have generated, you can generate the PNG, and you can see, open the PNG within the notebook, and you can see this was the workflow that we have seen earlier. And it's a very nice way of visualizing the steps of execution within your workflow. A great feature. Okay, so we have visualized the workflow. So now I am gonna ask Snakemake to dry run the workflow using the -n flag just to see how many jobs does it have. So you can see that there are total 20 jobs in total with all the individual jobs listed over here, and looking by the number of jobs in get SRA by accession, there are 3 samples that are involved. As we discussed, there's 1 test sample and 2 control samples. So all of that looks great. So now let's put up this workflow for execution, and then we'll go over and look at the Snakemake snake file. Okay, so for this, I am removing the -n flag, and here I'm going to introduce another flag called --course, which is basically to allow Snakemake for multi-threading, for parallelization. So in this case, uh, let me see how many cores do we have on this particular instance. So for that, I'm launching another terminal and using an application called htop. So I have 16 threads in this machine. So I'm going to use 14 and enter. So you can see that this particular Snakemake execution has started. Uh, Now let's look at the snake file and talk about some of the differences. You might notice that when we first ran our Snakemake workflow, we gave the target file that it was supposed to generate, like _a_b.txt. But notice how we didn't have to give the target file here, because the difference is that the first is uh, snake file is by default is the default file for Snakemake. So Snakemake by default will look for a file that is called snake file with a capital S in the folder that is located. Uh, Now, coming to why we did not have to define any target files, it is because that Snakemake by default targets the first rule of its workflow. So now if you see in the workflow, in the snake file, our first rule is this rule called rule all. So by convention, this rule all is what serves as a rule for aggregating the main outputs of the workflow. So here you can see in rule all, we are asking it to generate uh, all the output files that we want from this workflow. So in this case, by listing all the output files in your rule all, that is, these are the target files that you want to generate in your rule all, you don't have to specify them on the command line, and Snakemake by default will run the first rule. So in this rule, it will see that it has to generate the outputs generated by other rules. And it is important to understand here that in Snakemake, the logic is implicit and backwards. So based on what we asked Snakemake to create in this target rule called rule all, Snakemake will go back and check the dependencies of dependencies of the rules that are implicit and force the execution backwards. So what we are trying to say is, in the rule all, when we ask it to make counts.tsv, it will find the rule that it will help make counts.tsv. And in this case, it would be the generate count table. And you can see in the output, this is where we are generating the counts.tsv. And in order to generate counts.tsv, Snakemake will go force execute all the rules that it will help to generate count.tsv. So in this way, you don't have to specify it step by step in the order of execution. Rather, you just tell it what is the target file that you want to make, and Snakemake goes backwards and determines what are all the rules that need to be rerun, and it will execute the workflow for you in that way. So I hope you note, I hope you understand the difference here in this case, where with the rule all, where we are specifying the target files that we want to generate within the workflow in the rule all, and Snakemake will, based on all the targets within the rule all, go backwards and run all the rules that it will help to generate the final set of target files. Okay, so now if we look at our Snakemake workflow has finished, and you can see that a bunch of folders have been created. Here is the data folder with the raw data. Here is a folder with all the intermediate files, which is, remember we talked about preserving all the files through data transformation stages for the sake of reproducibility. And also the results folder contains the supplementary PDF file. And in this supplementary PDF file is a report that you would want to generate at the end of your research project, that captures all the information on what were the samples, what were the methods used, and also see the results. Here is the Snakemake workflow that was used to generate this particular analysis, and it also captures all the packages that were used for generating these. So this is tying back to the main theme of reproducibility that we were talking about, where you want to make sure that every step is documented. So I hope everyone was able to run the RNA sequencing Snakemake workflow. And before we move on, I do want to.
Satish, check your chat messages. I think there's an answer there for one of the earlier questions before we move on to other questions.
Yes. Oh, yeah, Nirav. Sorry, I don't see anything in my chat. Oh, that is for breaking the list. Oh, so Nirav.
Okay,
Oh, I answered the question about thousands of input files was to put the list of them in the config file. Right. Sure. And use a wildcard in the rules and to load that list. Sure. I would definitely agree with you, Daniel. And just as I was about to touch upon that. So in this particular snake file, in the beginning, just before the rule all, see how we have a config file where we are reading in config.yaml. And the config.yaml file is where you list out the different samples that are involved in your study, and maybe even different attributes. Like here, you can see the different genomes that we are downloading, the GFF files. So this is an efficient way of keeping your Snakemake workflow generalizable, such that you have your generalized RNA sequencing workflow, and you store information specific to any project in this config.yaml file, which will be read in by the snake file. So thank you, Daniel. Yeah, sure. Definitely having them in a config.yaml is, as in this case, as you can see in this particular snake file, is a very valuable thing to have, I would say. Okay, so just before, I do have a few more points to touch on. First thing is please, I would request everyone to take their time to finish the rest of the tutorial, because we don't have time to cover all the sections, features within the tutorial, and also all the other features of Snakemake. You can, there are quite some features depending on how you want to build out your workflow. So definitely I recommend you to finish the rest of the tutorial on your own. uh, I do want to uh, now just touch on two last things before we close. Is for the sake of reproducibility, we talked about, like, so this is an example of how you would want to organize your project, or how you would want to organize the analysis that you're doing within your project, where you have everything clearly marked out. As you can see, the code, data, intermediate results, everything is organized. Your config files, your samples are indicated in your config file. And if you see the environment.yaml file, here are all the tools that are currently installed using Conda in this particular app for execution for RNA sequencing analysis workflow. But this is a great way for aiding reproducibility, so that now if I want someone to replicate the same environment, all I need to do is just give them my environment.yaml file, and it should install or set up the same software execution environment for me, or for anyone else. And also the snake file. So these are some of the basics of how you would want to organize your project, and make sure, and some of the files that you want to have in your project, so that you're aiding in the reproducibility, such that all you need to, for someone to replicate this project, is to give them the config, the environment.yaml file, put them in a GitHub and share it, and people should be able to replicate the study with all this information. And on the last point, is something that is not part of this webinar, but I do want to show this image, which I thought was really nice. Is this So in the course, Tools for Research, designed by and folks at NBI Sweden, they have listed out these 6 tools as what you would want to have in your toolkit for reproducibility. Git for version control is where you track and back up your project history. Environment management through Conda, where you set up and manage your project environment. Workflow management using Snakemake, move from separate scripts to connected analysis. R Markdown for all your reports, connect your code, output and text and beautiful reports. You see the supplementary.pdf that was generated in the results file, that was using R Markdown. Jupyter notebooks for documenting all your exploratory analysis. Now remember, we have done all our analysis in this Jupyter notebook, which I will show you how to save those results into your discovery environment at the end. And also something about containerization with Docker and Singularity, where you not only just capture the whole operating system and make your project self-contained and distributable. So for people, or especially instructors who are interested in conducting a class where they make available such an app that we have used here today, just a bit of information is that this particular Snakemake Wise app is built using Docker, and it runs via a Jupyter Notebook interface. And using the environment.yaml file that we see here, I have built this app using Docker. I have updated the Docker container to contain these tools, but you can very well do it with whatever research tools that you have, and integrate them into the discovery environment, and make them available for your class to follow a particular tutorial, just as we did today. So if you are interested, please check out CyVerse Learning Institute material on how to add your app and make it available as a Wise app, such as either Jupyter Notebook or an R Studio. Okay, so final thing is how to save all these outputs and results onto your CyVerse data store. Now that we have finished all our analysis here and our results have been achieved, now you can close this tab and go back to CyVerse discovery environment. And if you bring up the analysis window by the analysis tab by clicking on the analysis tab, it should pop up that the current analysis that is running, and this is my notebook that is currently running. And now I wish to complete and save output and bring all the data that I have done by processing there into the CyVerse discovery environment so that you can share it with others, is where you find your analysis and click on the three dots. And there's an option that says complete and save outputs. So if you click on that, the notebook server will be shut down and all the data in your notebook is transferred to your CyVerse data store. So if you click on the analysis name, it will take you uh, to the folder with all the results that you have been saved from the notebook. So with that, I hope the webinar was helpful. And I hope you go out to build some very great workflows. If you do, please share with us, tweet, or send us an email. We'll be happy to look at the workflows. But with that, I wish you all good luck in snake making and stay safe. And thank you so much. And I will be happy to take questions.
Thank you, Satish. There aren't any new questions, but there are some really good comments, and I do appreciate everyone's good questions because it just brings out what different people are doing and where the fault problems are lying. So again, join us in a couple weeks for Noah Falgren's webinar. And with that, let's close the webinar and see you in a couple weeks.